Decoding the Future: Gene Selection in Unborn Children, a public discussion. Ever wondered if we're on the cusp of choosing our children's genes? Let's dive into the science, ethics, and realities of gene selection for unborn children, separating fact from fiction. This is a discussion about the future of humanity, and your voice matters. Genetics is the study of heredity and the variation of inherited characteristics. It's the blueprint of life, dictating everything from our eye color to our predisposition to certain diseases. Reproductive technologies like IVF are changing how we conceive and are opening doors to manipulating this blueprint. In vitro fertilization, IVF, is a process where eggs are fertilized by sperm outside the body. This is a common procedure for couples struggling with infertility. But IVF also provides an opportunity to examine the genetic makeup of embryos before they are implanted. Pre-implantation genetic diagnosis, PGD, is a technique used alongside IVF. It allows doctors to screen embryos for specific genetic disorders before implantation. This means selecting embryos that are free from certain diseases, offering a chance for a healthy start. PGD is primarily a screening tool. It identifies existing genetic conditions but doesn't alter the genes themselves. It's like choosing the best apple from a basket, rather than changing the apple's DNA. PGD is used to detect conditions like cystic fibrosis, Huntington's disease, and Down syndrome. It helps parents make informed decisions about which embryos to implant, reducing the risk of passing on inherited diseases. CRISPR-Cas9 is a revolutionary gene editing tool. It allows scientists to precisely alter DNA sequences, offering the potential to correct genetic defects. However, its application in human embryos is fraught with ethical concerns. The CRISPR technology works like a precise pair of scissors, cutting and pasting DNA sequences. This allows scientists to remove faulty genes or insert new ones, potentially curing genetic diseases. In 2018, a Chinese scientist used CRISPR on human embryos, resulting in the birth of genetically edited babies. This sparked global outrage and highlighted the ethical risks of germline editing. The scientific community largely condemned this experiment due to safety concerns and ethical violations. The prospect of gene editing raises serious ethical questions. Are we playing God? What are the long-term consequences of altering the human genome? The idea of designer babies, selecting for traits like intelligence or appearance, is particularly controversial. One major concern is the potential for off-target effects, where CRISPR edits the wrong genes. This could lead to unintended mutations and health problems. The long-term effects of gene editing on future generations are also unknown. The ethical debate also revolves around equity and access. If gene editing becomes available, will it only be accessible to the wealthy? This could exacerbate existing social inequalities, creating a genetic divide. The legal landscape surrounding gene selection and editing is complex and varies widely across countries. Some nations allow PGD for medical reasons, but prohibit gene editing. Others have stricter regulations, banning any manipulation of the human germline. In the United States, there is no federal law specifically banning gene editing in embryos. However, the FDA prohibits the use of federal funds for research involving the creation of human embryos for research purposes. Many European countries have strict laws against germline editing. Germany, France, and Italy, for example, prohibit any alteration of the human germline. These regulations reflect deep ethical concerns about the potential misuse of gene editing technologies. Traits like intelligence, height, and personality are influenced by hundreds of genes and environmental factors. Selecting for these traits is incredibly complex and may not be feasible with current technology. These traits are known as polygenic traits, meaning they are controlled by multiple genes. Each gene contributes a small effect, making it difficult to predict the outcome of selecting for these traits. Environmental factors such as nutrition, education, and social environment also play a significant role in shaping these traits. Even if we could select for certain genes, the environment could still have a major impact. Germline editing involves making changes to sperm, eggs, or embryos that can be passed on to future generations. Somatic cell editing, on the other hand, affects only the individual being treated. Germline editing is far more controversial due to its heritable nature. Somatic cell editing is similar to traditional gene therapy. It targets specific cells in the body to correct a genetic defect. These changes are not passed on to future generations. Germline editing, however, alters the DNA of reproductive cells. This means that any changes made will be inherited by future generations. This raises concerns about unintended consequences and the potential for altering the human gene pool. While gene editing in embryos is still largely experimental, gene therapies for treating existing conditions are becoming more common. These therapies often involve delivering a functional gene to replace a faulty one, offering hope for patients with genetic diseases. Gene therapies are being developed for a wide range of conditions, including spinal muscular atrophy, hemophilia, and certain types of cancer. These therapies have shown promising results in clinical trials, offering the potential for long-term cures. In 2023, several gene therapies were approved for use in the United States and Europe. These approvals mark a significant milestone in the field of gene therapy, paving the way for more widespread use of these treatments. Even if gene selection and editing technologies become more advanced, accessibility and affordability remain major concerns. These technologies are likely to be expensive, potentially creating a divide between those who can afford them and those who cannot. The cost of IVF and PGD is already a barrier for many couples. Gene editing would likely add to these costs, making it even more difficult for low-income families to access these technologies. This raises questions about equity and social justice. Should these technologies be available to everyone, regardless of their ability to pay? Or will they become a luxury available only to the wealthy? It's important to distinguish between selecting genes, like choosing between existing embryos, and editing genes, changing the DNA. PGD is a form of selection, while CRISPR is a form of editing. The ethical implications of each are different. Selection involves choosing the best available option from a set of existing possibilities. Editing involves altering the options themselves. This distinction is crucial when considering the ethical implications of these technologies. PGD is generally considered less controversial than gene editing because it does not involve altering the human genome. However, even PGD raises ethical questions about the selection of embryos based on certain traits. Besides PGD and CRISPR, other technologies are being developed to address genetic diseases. Mitochondrial replacement therapy, for example, can prevent mitochondrial diseases from being passed on to children. Mitochondrial replacement therapy involves replacing the mother's mitochondria with healthy mitochondria from a donor egg. This prevents the child from inheriting mitochondrial diseases, which can cause severe health problems. Polygenic risk scoring is another technology that assesses the risk of certain diseases based on multiple genes. This can help individuals make informed decisions about their health and lifestyle. Most experts agree that using PGD for serious genetic conditions is acceptable. However, using it for enhancement or non-medical traits is not. The technology for selecting embryos based on non-disease traits is not very advanced because those traits are complex and influenced by many genes. The scientific community generally supports the use of PGD to prevent serious genetic diseases. However, there is widespread concern about using these technologies for non-medical purposes. Many scientists and ethicists believe that using gene selection or editing for enhancement purposes could lead to unintended consequences and exacerbate social inequalities. Gene editing for unborn individuals, particularly through in utero or prenatal approaches, represents a rapidly evolving field with potential to prevent or treat genetic diseases before birth. This approach offers unique advantages, but also raises significant ethical and safety concerns. In utero, gene editing involves delivering gene editing tools directly to the fetus. This could potentially correct genetic defects before birth, preventing the development of serious health problems. Studies in mice have shown that in utero, gene editing can correct genes linked to conditions like hereditary tyrosinemia type 1 and Angelman syndrome. However, these studies are still in the early stages and much more research is needed. Fetal development offers benefits like immune tolerance to vectors, high cell proliferation rates, and access to organs, e.g., the brain, before the blood-brain barrier fully forms. Early intervention may prevent irreversible damage from diseases with perinatal onset. The fetal immune system is more tolerant of foreign substances, making it easier to deliver gene editing tools without triggering an immune response. Fetal cells also divide rapidly, allowing edited cells to expand and replace faulty cells. Early intervention can prevent irreversible damage from diseases that begin before birth. This could significantly improve the health and quality of life for affected individuals. Germline versus somatic editing, maternal-fetal dynamics, equity and access, and designer baby fears are all part of the ethical and social concerns. These issues require careful consideration and open discussion. Most prenatal editing targets somatic cells, but accidental germline edits could transmit changes to future generations. This raises unresolved questions about long-term effects and intergenerational consent. Editing a fetus inherently involves the pregnant person's health risks, potentially linking the technology to debates about abortion access and maternal autonomy. Equity and access are also major concerns, as emerging therapies like CRISPR-based treatments for sickle cell disease highlight disparities in healthcare access. Public opinion on gene editing is divided. 52% of Americans oppose gene editing to reduce disease risk in babies, while 46% support it. Approval rises when the goal is direct health benefits, 72%. The legal landscape is also complex, with varying regulations across countries. The U.S. prohibits federal funding for human germline editing research and implantation of edited embryos. However, privately funded research continues, albeit without clinical trials. In 2018, a Chinese scientist controversially edited embryos to confer HIV resistance, resulting in jail time and international condemnation. This event highlighted the need for clear ethical guidelines and regulations. Research aims to refine delivery systems, expand targets to monogenic disorders like cystic fibrosis, and address ethical challenges through inclusive policymaking. While prenatal gene editing holds transformative potential, its translation to humans requires rigorous safety validation and broad societal consensus. Scientists are working to develop safer and more efficient delivery systems for gene editing tools. They are also exploring the potential of gene editing to treat a wider range of genetic diseases. Addressing the ethical challenges of gene editing requires open and inclusive discussions involving scientists, ethicists, policymakers, and the public. This will help ensure that these technologies are used responsibly and for the benefit of all. Prenatal CRISPR-Cas9 and base editing corrected the HPD gene in mice with hereditary tyrosinemia type 1, HT1, rescuing lethal outcomes and maintaining edited liver cells for up to 3 months. Treated mice outperformed those on standard drug therapy, nitisinone, in survival and liver function. Lipid nanoparticles, LNPs, delivered mRNA encoding Cas9 into fetal mouse brains, editing genes linked to Angelman syndrome. This approach leveraged fetal immune tolerance and early blood-brain barrier permeability to halt disease progression. Nanoparticle-delivered gene editing tools achieved a 6 to 10% correction rate in V. thalassemia mouse models after a single in utero treatment, outperforming postnatal interventions. These results demonstrate the potential of in utero gene editing to treat a variety of genetic diseases. Fetal exposure to editing tools, e.g., Cas9, avoids antibody formation, enabling repeat postnatal treatments if needed. Fetal progenitor cells' rapid division allows edited cells to expand, enhancing long-term efficacy. It also targets diseases like neuronopathic Gaucher or surfactant protein C deficiency, where pathology begins prenatally. The fetal immune system is more tolerant of foreign substances, making it easier to deliver gene editing tools without triggering an immune response. Fetal cells also divide rapidly, allowing edited cells to expand and replace faulty cells. Early intervention can prevent irreversible damage from diseases that begin before birth. This could significantly improve the health and quality of life for affected individuals. Editing rates vary, e.g., 15% in liver cells, though even low rates can be therapeutic if edited cells have survival advantages. No germline editing was detected in animal studies, but off-target effects and long-term consequences remain unresolved. No human trials exist yet, and scaling delivery methods, e.g. LNPs, for larger fetuses requires innovation. Editing efficiency is a major challenge. Even if gene editing tools can be delivered to the fetus, they may not be able to correct the faulty gene in all cells. Safety is also a major concern. Off-target effects, where gene editing tools edit the wrong genes, could lead to unintended consequences. The long-term effects of in utero gene editing are also unknown. Accidental edits in reproductive cells could affect future generations, necessitating stringent oversight. High costs and uneven access to prenatal diagnostics may limit availability for marginalized populations. The risk of germline editing is a major ethical concern. If gene editing tools accidentally edit reproductive cells, the changes could be passed on to future generations. Equity is also a major concern. If in utero gene editing becomes available, it is likely to be expensive, potentially creating a divide between those who can afford it and those who cannot. The fetal immune system is tolerogenic, meaning it is less likely to mount inflammatory or neutralizing responses to viral vectors or transgene products. This allows sustained expression of therapeutic genes without immune clearance. Up to 30 to 60% of adults have antibodies to AAV from natural infections, which neutralize gene therapy vectors and block efficacy. Similarly, pre-existing T cells targeting Cas9 from bacterial exposure can attack gene-edited cells. Prenatal delivery of human factor IX, HFIX, in mice induced lifelong tolerance, enabling repeat postnatal doses without antibody formation. Adult mice injected with AAV1.HFIX developed anti-HFEX antibodies, whereas prenatal treated mice did not. Prenatal CRISPR editing in HT1 mice achieved 15% liver correction, with edited cells persisting for months. Postnatal editing in the same model led to anti-Cas9 antibodies and cell loss. Prenatal AAV delivery in sheep induced tolerance to HFIX, while postnatal delivery triggered antibody formation. These examples highlight the advantages of prenatal gene therapy in terms of immune tolerance. Prenatal gene therapy leverages fetal immune tolerance to enhance efficacy and enable repeat treatments, whereas postnatal therapy faces significant immune barriers. However, prenatal approaches require careful timing to avoid maternal antibody transfer and rigorous safety checks to prevent germline editing. The science for selecting genes of an unborn child is partially available, primarily through existing technologies like PGD, but with significant limitations and ethical considerations. Gene editing technologies like CRISPR hold promise, but are not yet safe or approved for use in human embryos. Selecting for non-medical traits is technically challenging and ethically controversial. Legal restrictions vary by country, and the feasibility depends on the specific trait in question. Most applications are currently limited to preventing serious genetic disorders rather than selecting for desired characteristics. The future of gene selection and editing is being shaped now. We need a broad societal conversation to ensure these technologies are used responsibly and ethically. What are your thoughts? What are your concerns? Take action now. Share this discussion. Help spread awareness and encourage others to join the conversation. Research further. Explore reputable sources to deepen your understanding of genetics and gene editing. Contact your representatives. Advocate for responsible regulation and ethical guidelines. Leave a comment. Share your thoughts and questions below. Let's shape the future together.